Hello and welcome to Biology 240. This is microbiology and this is chapter 3. We are now looking at the cell. And remember, these lectures are meant to supplement and highlight what you read in the book and then during class discussions. So please come to discussions and we'll look at this. But what I want to look at here today in the biology of the cell is we're going to review a little bit of what's going on with spontaneous generation. This is the idea that will not die, modern cell theory, and then we're going to look at characterization of what prokaryotes are and then what the characteristics of eukaryotes are. Again, remember that microorganisms are things that at some point in their life cycle, that is kind of when they are born to when they die, that they are less than 0.1 millimeter across. They can be multicellular, but these are kind of very small. They come in many, many, many shapes. Okay. What we want to talk a little bit about is kind of where life comes from. And you've heard all this thing about spontaneous generation. And so what this comes about is you get this idea of spontaneous generation is when you don't know where things kind of come from. They can just kind of come out of thin air, and maybe it's magic, and maybe it's something that you think when you're a kid or kind of something like that. And so what we tend to see in science is, like, can we have a relatively simple experiment that would prove that this doesn't happen? And so here's an example of Francisco Redi that did this, a long time ago. And basically, you take a piece of meat, and boom, you have spontaneous generation. These maggots occur out of the meat, but if you seal the meat, there's nothing happens in there. And then people are like, oh, the force can't get in there. And then if you do that, you put some cheesecloth on top of this. You can see the magnets and the flies up in there, and your meat won't do it. So what we tend to see, and what we see a lot, is that we kind of look for these experiments or these demonstrations that would tell us kind of what's going on in nature. And so we're going to go through this. The prize that actually put all this is again, a lot of people, even when you say, oh, well, maggots don't come out of thin air, but they're like bacteria can come out of thin air. And the case of Louis Pasteur showed this, and he actually won a prize on this. And he had this long swan neck flask, and he showed that you can boil a media inside of this flask, and then you can bend the neck of the flask, and if materials can't get back in there, then they don't. Then nothing grows in there. And if you kind of break off the top of this flask, that stuff will still kind of grow inside of there. Again, remember your timelines that you guys all made. And so Robert Hooke was one of them, and we do a whole bunch of different things. Again, scientific theories sometimes do not catch on. And Vickrell and a lot of these guys are talking that cells only come from cells. We don't have spontaneous generation. That came up. Wallen in 1920s had this idea of where the organelles in eukaryotic cells came from. And so, again, we were looking at how did eukaryotic cells occur, and that we think there's this proto-eukaryote. This proto-eukaryote then developed a nucleus and the endoplasmic reticulum, and that we think it took in some aerobic bacterium and possibly some photocytic blue-green algae, and that's what gave us our kind of modern eukaryotic cells. And I think Wallen believed that he was able to cultivate some of these, which is kind of necessarily the case. But we have this thing where it's a good idea, and it just kind of really didn't catch on for many, many years. Ignaz Semmelweis, who I love saying his name, basically came up with the importance of hand washing. And again, when he did this, a lot of people laughed at him, and they didn't want to wash their hands. And people for a long time had laughed at kind of washing their hands. But it really showed the case, and there's a lot of disease and death and destruction that could be prevented by just washing your hands. And this kind of reminds me of just this past year and us not wearing a mask. And actually, I was one of them too, of kind of poo-pooing that we really don't need to wear masks. And you know what? Quite frankly, it is a really good it's a really good thing to do. Review your timelines. Again, Joseph Lister, Robert Cox. These are all kind of mentors or the start of what we call microbiology. Know the parts of prokaryotic organisms. Just like you're going to know the parts of the eukaryotic organisms, I'm going to ask you to compare and contrast them. And so a typical prokaryotic cell contains a cell membrane, chromosomal DNA, ribosomes, and a cell wall. Some of them also possess villi, fimbriae, and capsules. and we will talk about all of these and kind of what they are. Again, microorganisms for the most part don't really have a big difference between them. And so, again, you can look at 3 different trees and they can all have kind of different shapes in general. And bacteria, it's basically you don't have, you have cocci, which are round, bacilli, which are rods, and vibrio, which are kind of spiral-like. And so, again, know what some of those common shapes are for bacteria. We're going to look a little bit at the physical properties of cells and their parts. And basically what you have is bacteria when they're in a solution. It can be what we call isotonic, which is the same. And so things leak out of the cell as fast as they kind of leak into the cell, in which case the cell will maintain. And so again, we're looking at a 20% solute concentration, 80% water. So solute is what is dissolved into a solvent, sugar into water, salt into water. Again, when we have a high solute concentration, what happens, the water leaks out of the interior, and this is what we call a hypertonic solution. In a hypotonic solution, you actually have water going into your microorganism, and this can have a case, and we use this a lot during growth of microorganisms. And so again, when we go to the hospital, you need to have an isotonic solution when they put it in you, or otherwise you will have an effect. And so what happens to your cells when this happens? In an isotonic solution, these concentrations are going to remain the same. In a hypertonic solution, the water particles move out of the cell, and we'd have what we call crenation inside of there. And so here's the cell losing some of its liquid. In a hypotonic solution, the cell would gain some of this liquid. And if you can see here, if you have a cell wall, cell walls are really helpful to a cell because they actually prevent the cell from actually exploding when that happens. Cells actually have DNA, and this DNA is in what we call a nuclear region. We call it a nucleoid. It's not necessarily a nucleus inside of there, and so that's where your DNA is going to be. The ribosomes are slightly different, and we have what we call 70S ribosomes, and that is they weigh a little bit different than ribosomes in eukaryotic organisms. And so, this, kind of came up in the 70s of kind of what are these kind of differences. Prokaryotic cells have various types of what we call inclusions, inclusion bodies. They can be hydroxybutyrate. We see a number of these magnesomes. We see a number of these things underneath the microscope. We can view them. Bacteria can also go through what we call a development process, and we talked about that in chlamydia, where it recognizes where it is. A lot of your bacilli can produce a spore. A spore is a kind of a resting structure, and spores can actually live for a very long time and dry and exposed to UV light, etc. And so here are these kind of vegetative cells with the spores inside of them, and when kind of things go wrong, these spores are able to survive, and spores are able to survive things like boiling water, etc. You do need to know kind of some of the properties of the plasma membrane. The plasma membrane is a phospholipid bilayer. It's a lot like a soap bubble. Within this plasma membrane, you have a number of proteins that are attached, and then you have a glycolipid that's attached on the outside. And so here's an example of how things can kind of move across a plasma membrane. And so again, what we tend to look a lot at is what these things that we call concentration gradients. You have a high concentration of sugar, so this is a 6-carbon ring, it's probably sugar, glucose. You have a high concentration of sugar on the outside, and what can happen is that material can diffuse through the plasma membrane. Now it's difficult, it depends a lot on the properties of that solute, of whether it can get through a plasma membrane or not. What we tend to see a lot more is that bacteria and other living organisms have these transport channels, and so these protein transport channel or water channel, and what this allows, it's a little hole inside of the plasma membrane, and it allows materials to kind of get through. This larger material probably can't get through, the smaller materials can do. This is what we call facilitated diffusion. And so diffusion is movement of substance from a high concentration gradient to a low concentration. Facilitated diffusion, you basically have a protein that facilitates, helps it along. In active transport, what you're doing is you're moving things against a concentration gradient, and you're actually using energy harvested from the hydrolysis of ATP to make ADP and an organic phosphate. And we use this to kind of gain some energy, and this is pumping. And you know what? This is a definition of life. When things are alive, they are pumping things across a gradient. When things are no longer alive, things are equilibrium, and this does no longer happen. Now, you may have heard a lot about peptidoglycan. This is composed of polymers of alternating NAG-NAM subunits, which are N-acetyl acid. These are held together by peptide bonds, and these are the structure that actually allow the membrane to happen. And in your gram-positives and your gram-negatives, you're going to have a very similar structure of this, but what we tend to see in our gram-positives is we have a lot of this peptidoglycan cell wall. In your gram-negatives, you tend to have a little bit of this cell wall, one layer. And in your gram-negatives, you tend to have what we call two membranes. You have an outer membrane and an inner membrane. And in your gram-positives, you only have the one membrane in there surrounded by this very thick wall. And that's why we tend to see the difference in staining between those. And so, again, here's an example of in a acid-fast bacteria, we may have on the outer surface these kind of mycoic acids, which are these very lipid-like structures that are outside of it. And so that's one of the reasons why these acid-fast bacteria tend to be variable. On our gram-negatives, we have a very reactive substance called a lipopolysaccharide, an LPS. And an LPS is basically a pyrogen, and it actually causes fevers and things like that as well. And so you can actually have a material that's sterilized, but if it's contaminated with lipopolysaccharide, it'll actually cause fevers and things like that. Protective coatings include capsules, or as a type of glycocalyx on the outside. And so here we have this negative staining, and this kind of little white area seen here is that capsule. Again, one of the reasons why we have to do this kind of staining is these capsules tend to not have a charge, and it really won't, either an acidic or a basic stain will not stick to those. Pili are actually these channels that bacteria can form to one another. They can have sex with it. They can communicate a number of different ways. And then flagella is bacteria actually have a way of moving. They have this kind of whip-like structure that allows them to move. And basically it's a rotary motor. And what happens is this flagella kind of whips around and allows it to move. And again, we have what are called monotrichous, in which case you have one flagella, amphitrichous, where you have two flagella, and peritrichous, where you have flagella all over it, and this allows you to go. What we tend to see is that when these flagella rotate clockwise, they kind of separate and the bacteria tumble. When you rotate counterclockwise, the flagella all come together and they rotate and they propel the bacteria forward. And this clockwise rotation is what we call tumbling, and the running is the counterclockwise. And what we see is bacteria can actually sense a gradient. And so if they're moving in a sugar solution, they will actually go towards the higher sugar solution because they will run longer under those conditions. Now, if the conditions are opposite, it's unfavorable, and let's say it's alcohol or something that will kill the bacteria, it will run more in the opposite direction. Woo! Does everyone get the prokaryotic cell? For the eukaryotic cell, it's a little bit different. In the eukaryotic cell, we have a lot of these kind of what we call organelles, these membrane-bound organelles. And these can be, a eukaryotic organism can be single-celled inside of there. Again, they come in a variety of shapes, and we've seen this kind of shapes before. Eukaryotic cells have a well-defined nucleus, and this is an example of mammalian lung cell, which we have a large, we have a structure called the nuclear lamella, and so there's a membrane that surrounds the nucleus that controls what gets into the nucleus, and that actually controls what gets out of the nucleus as well. Right next to the nucleus is the endoplasmic reticulum. But in the nucleus itself, you have an area where actually the ribosomes are actually, the rRNA is transcribed, and then the RNA is partially assembled inside of there. So you have these ribosomes that are coming about. I liken the endoplasmic reticulum as kind of like a kind of a manufacturing assembly line. And so you actually have rough endoplasmic reticulum, and then towards the outer portion you have the smooth endoplasmic reticulum, and then this material will go into the Golgi. And so again, when a cell wants to make something, it will make a messenger RNA. This messenger RNA will get transcribed and then translated into a protein inside the endoplasmic reticulum or going into the membrane. So the rough endoplasmic reticulum is studded with ribosomes, and that's where a lot of the production of protein occurs. And the Golgi apparatus is where you see that those proteins are modified and then they're packaged to kind of get to the end. This is a transmission electron microscope showing you another structure that we see in the cells. These organelles are independent of the Golgi complex. That's not to say that they aren't made by this material, but in the Golgi complex, what you're doing is you're having materials, information that's converted into a messenger RNA inside the nucleus. As this moves out into the cell, it is actually made into a product, and then it needs to go where it needs to go inside of the product. We have these other organelles that are actually called things like peroxisomes, that where we see the cell does different activities. Another important part of the cell is just not a bag of liquid. We actually have what we call cytoskeleton inside of there, and so you have a number of fibers, and then so you have tubulin, we have actin, we have keratin fiber subunits that actually make up the skeleton. The actin allows for movement inside the cells. The intermediate fibers allows for the structure of the cells, and then the microtubules allow us to kind of change the overall shape of the cells and for some movement. The centrosome is controlled with two centrioles positioned at right angles to one another, and this allows for movement of the chromosomes. Another organelle that we tend to not see, again, this is not part of the endomembrane complex, and basically what we're talking about are things like the mitochondria or the chloroplast. The mitochondria, we think, is a microorganism that was captured by it, because again, it has its own DNA, it has its own set of ribosomes, and these ribosomes are 70S ribosomes like we see in other organisms, and the mitochondria are responsible for the production of adenosine triphosphate, and again, so are the chloroplasts, and again, we'll talk about those a little bit later. A phospholipid bilayer, so membranes also occur in eukaryotic cells, and they're very similar to the prokaryotic cells as well. One of the big differences that we have in, again, eukaryotic cells is this kind of movement of materials into and out, and we have this process that we call phagocytosis, where a piece of the membrane pinches off around an organism, and it will form a vacuole on that organism. The macrophages tend to do this a lot, and we'll talk about those a lot. We actually have pinocytosis, and we have receptor-mediated endocytosis, where things are kind of taken into the cell by the cell structures. Outside the cell of a eukaryotic organism, we can have something that's similar to the glycocalyx of the bacteria, and we have this extracellular matrix. This extracellular matrix is the glue that actually holds the cells together. It's this collagen that holds the cells together, and we tend to see that. And so what I want you to be able to do is, here's a cartoon of a prokaryote. You need to be able to fill it out, tell me what the parts are, and tell me what they do. Again, here's a eukaryotic organism. You need to be able to fill those parts out and kind of tell me what to do. This has been Nick Cap, and this is your kind of few moments looking at the complexity of both prokaryotic and eukaryotic organisms. Thank you very much for listening to me. If you have any questions, come to the lecture. We will have a discussion on Wednesday, and you could also move on to the discussion board as well. Thank you very much.